Hi, I'm Dr. Amy Turner and welcome to Proteintech's podcast, the platform where we discuss the big questions of the day that help us to understand the book of life. Our guest today is Dr. Iván Godinez, Director of Product Management at Proteintech Genomics. Iván is a leading expert in single-cell proteogenomics and has over 10 years of experience developing research tools for the multiomics space. Beyond his scientific career, he has a plethora of hobbies, not least wrestling, piano playing, guitar playing, and even being a pilot. So, Iván, welcome to today's podcast. It's a pleasure to have you here.
Well, I'm excited to be here as well.
Amazing. So, Iván, you've had a wonderfully successful career since your PhD, which was performed at UC Davis in the States. And this was in immunology with a specific focus on microbiology. Is that right?
Yeah. We were interested, or I was interested in what's called host pathogen interactions. You know, there's a lot of people who study bacteria or viruses, and then there's people who study the organisms that are infected, humans or other animals. And I was really interested at the interface. What happens to the organism in the presence of a bacterial pathogen, in this case, Salmonella.
Amazing. So in that space, obviously you've come to genomics at the moment, but this was very much a microbiology area.
Yeah, it was a very, very, different space, different time back then. You know, the closest thing we had, which is genomics, was qPCR or qRT-PCR, where you could measure several genes or, dozens of genes from tissue, which was really exciting because you could get a system level view of what's happening to the host in the presence of bacteria. So you see what genes are being turned on and what are the implications of those genes being turned on in the case of infection, which is, talking to my PI at the time, he's like, well, we would identify one bacterial gene, sequence it, and that was your PhD. So it was a novel time and very exciting time to do your PhD in that space.
Definitely. How much do you think that space has grown in the last sort of 20 years?
Quite a bit. I mean, infectious disease is always around, whether it's COVID or God knows, Mpox and all these other things. Understanding how the host, in this case humans, responds and what are some of the factors that can impact how one does during these infections is always going to be important. Mm-hmm. So the tools, whether it's single cell RNAseq or other genomics applications, are always going to lend a hand in that space. So I think people, whether you're studying bacteria, it has grown into like microbial population. So doing population-wide microbial genetics, doing, single cell on the host side of things. So it's definitely kept up. And you see it impacting the research and novel insights, new therapeutics. So it's definitely kept up and it's very interesting. Definitely.
Amazing. So during your PhD, was there any specific kind of big discoveries that you made?
I see. I was quite lucky in my PhD. I always say this, God loves fools, and I'm one of his favorite fools. And I walk into these situations and things just seem to work out. So I start my PhD program with Andreas Boimler at UC Davis, and he's like this awesome Salmonella guy. And they're talking about how the host responds. And I just say, why don't we just knock down these host cells? And at the time, they're T cells. I mean, they're still T cells. So I made this simple suggestion, and that led to a very elegant experiment, and it had very fruitful results. And from those results begat more results. And within a couple of years, I published 3 papers. And This was, you just lock in, right, being at the right time, the right situation with the right question. Mm-hmm. So I think the novelty to my project was specifically looking at at the time recently discovered what's called an axis of inflammation. So it's a series of events that occur within the host that leads to a disease, right? In this case, we're studying diarrhea,
right?
Induced by Salmonella.
And who doesn't love diarrhea?
Yeah, who doesn't? We've all had it, whether you like it or not. Yeah, and Salmonella is particularly interesting because it's transmitted via the fecal-oral route. So whether you like it or not, we've all had someone else's poop in our mouth. Got diarrhea from it. For most folks, you get sick, you spend maybe a few hours on the toilet and you get over it. You don't even realize you're sick. Some people, obviously there's varying degrees to what that happens. But in a in the population of people who had fairly advanced HIV disease or AIDS, when they got infected with Salmonella, the disease looked very different. They didn't get diarrhea. They got a disease that looked more like typhoid fever.
Wow.
Which kind of looks like a low-grade viral infection. And unfortunately, these patients would pass away because the bacteria moved from the intestinal lumen, intestinal tissue, into the organs, cause systemic failure and death. So my PhD was all about, well, what is it on the host side, the human side, that helps to control that infection? Why does the Salmonella infection in healthy human beings look the way it is? Meaning you get diarrhea. In the way the bacteria works, it comes in, it actually starts the diarrhea on purpose. That leads to the cultivation of an ecological niche that it has exquisitely evolved to take advantage of. And your normal host bacteria, they all die off. So it comes in, causes diarrhea, ruins the neighborhood, eats all that's left over, and then leaves and infects someone else. So that's what's supposed to happen. And that's what most of us, so basically what I figured out or helped, and obviously this was a big group effort and stuff with other scientists, was the role of a specific type of immune cell called a T cell in orchestrating that response to Salmonella. And yeah, so all my papers were all around, depleting different T cell populations in the gut, looking at the role of certain pro-inflammatory cytokines and discovered these two cytokines. I didn't discover the cytokines, but I helped elucidate the role that two cytokines, IL-23 and IL-17, had on coordinating this diarrhea response, inflammatory response. And interestingly enough, these cytokines are now therapeutic targets for like Crohn's disease. And so, you can now get an injection that stops your IL-17 signaling or depresses it so that you don't get this constant inflammatory state in your gut.
Wow, that's a big societal impact then from this one.
Yeah, from this one thing. I mean, I didn't do all that, but, I studied a little bit of it and, did a small contribution there.
Amazing. So after your PhD, you didn't do a postdoc, you transferred into industry where you became a sort of technical specialist. And this is where you were introduced more into the world of genomics is what it was at the time.
Yeah.
Maybe for our listeners, you could talk about what genomics is at a very layman's terms, if that's possible.
It's a bit of a loaded question, but yes, I can give it a shot. So, we're all made up of cells, right? For humans, I think it's estimated something like 37 trillion individual cells.
I think I heard somewhere that there's more cells in the body than stars in the sky, but I feel like that's
in the galaxy.
In the galaxy,
yeah.
Okay.
Yeah, there's a lot of cells, right? And each one of those cells has a copy of your genome. And the genome is essentially the cookbook to make you, the instructions. And genomics is all about, one, obtaining the sequence of your genome and helping and inferring from that or deducing from that the role that individual genes or parts of the genome have on human health. But this obviously applies to other animal models, other species as well. So I joined this company, Illumina, which is now a behemoth in sequencing. But when I started, it was a smaller company. And it was, honestly, I didn't want to work there. If I'm being honest. I was looking for postdocs and just really didn't have the desire to be in a wet lab anymore. So I went and found this job as a technical support scientist doing genomics. And at that time, I had this dreary view of technical support, right? Just rows and rows of sad people answering phone calls and just trying to get through everyone's complaints. And actually, I was happy to discover that wasn't the case. These very intelligent, amazing scientists would call and ask me for help with their sequencing instruments. So they're trying to, whether they were sequencing tumor cells or healthy cells or animal cells, their machines would break or they'd need help designing their experiments. And that's what I did. So I got to talk shop all day and solve people's problems, which was incredibly fulfilling, which was nice because I got to, keep my feet in science, but not do any of the actual
you didn't have to go
lab.
in at the weekend and deal with infected cells and that kind of thing.
That's right. Yeah. And I learned a lot, from it about genetics, about genomics, where the field was going. And I segued from more technical roles into more like product management, which is more about understanding what people need and helping guide organizations into providing for those needs, whether it's a specific type of instrument or a new reagent, which was really cool because I still got to talk to a lot of scientists, but it was, I wasn't solving immediate problems. I was solving more systemic problems, and which actually I'm still doing now and I really love.
Do you feel like at that time it was the sort of start of the boom of omics research? Obviously, it's not just genomics. You've got transcriptomics, you've got obviously proteomics, metabolomics, everything.
I mean, the advent of genomics, right? And there's different phases of it, right? We've sequenced genomes for a long time, whether it was I think the Haemophilus influenza genome was one of the first ones, and some viruses and stuff like that. But I got to join this company when it had these instruments that could sequence maybe half a genome, a quarter of it at a time. And by the time I left, I was supporting instruments that could sequence 50 or so whole human genomes in 48 hours at 30x coverage. It was just a phenomenal change and boom. So it was really fun being part of that and seeing it happen. And genomics is the kind of an inflection point for a lot of things. And now if you look at the life sciences, a lot of the assays, a lot of the technology is based on that core genomic technology of sequencing. So it was a privilege, to work there and learn and see. And now I've carried that kind of torch, and now I'm doing single cell, genomics or single cell transcriptomics, proteogenomics, and it's all based on that knowledge I got from the sequencing space.
Amazing. So taking it back to kind of where it all began, take me back to before your PhD in the early days. What were your major influences that made you want to be a scientist?
I was always a really curious kid. And I can't say there was ever one thing that ever really called me to it. I always liked biology, mostly because I was really good at memorizing stuff in it. And it was one of the classes that I actually did well in. And but the thing that really caught my attention were bacteria. Not so much from they're cool and neat, more oh, wow, these things can have the potential to decimate whole populations, right? Learning about the plague, learning about tuberculosis and these diseases, and they just piqued my interest. And, one of the areas I was initially interested in was the antibiotic resistance.
Yeah.
I was kind of a nervous kid, and I just remember thinking what's going to happen when, these drugs don't work anymore, right? And if you look at the history, infectious disease was the leading cause of death for most of humanity's existence. And right now we're in a novel time where it's not.
Yeah.
But, it's just a matter of time before that changes. So that idea is what got me into microbiology. And then I started doing microbiology and realized, I don't actually like doing microbiology, but I like biology. I like doing, I like the immune system. And again, it was one of those things that just kind of came naturally to me, and I enjoyed, and it blossomed into my PhD. And that's how I got into it.
Amazing. So you were at Illumina, you've done your technical specialty, you've met a accumulation of scientists that you've helped solve their problems. You then moved on to even bigger players in the market, and you've now come to Proteintech as the director of product management for Proteintech Genomics. Can you talk about the single cell analysis work that you're interested in, and how that kind of fits into the life scientist sort of research at the moment? How does it impact?
Yeah. So, to start, I'm working on a presentation for a conference I'm visiting, and putting a slide together, what sort of along these lines. And I was reading this paper, and it was a paper describing what's known as the human cell atlas. And it's driven by these brilliant scientists. The primary author on this paper was this brilliant scientist, Aviv Regev, who's from MIT, and now she's a head honcho at Genentech. And she wrote this line, and it really resonated with me. And it's basically the quote, and I'm paraphrasing here, was something along the lines like, to understand human biology, we must know ourselves, because everything we see at the organismal level, whether it's humans or birds or whatever, is really a reflection of what's going on at the cellular level. Like the cells are the interface at which life goes from being an amalgamation of complex molecules to being what it is now, right? So it always fascinated me, especially when I learned about single cell and like seeing like, oh, wow, you have, hundreds of thousand cells and you can classify them all in one experiment. And part of my background is in flow cytometry, which is a type of single cell analysis, but it's limited in that you can evaluate, maybe a few dozen proteins if you're really good at it and you have the right instruments at a time. But most people were, looking at maybe 14, 12, 10 proteins, maybe less at a time. So I immediately just saw the value in that. Like, oh, wow, you can do, 3 years of flow cytometry work in a couple experiments. And I remember thinking like, wow, I wish I had this during my PhD. So yeah, I got an opportunity while I was at Illumina. I got a phone call saying, hey, we're looking for a product manager that knows sequencing pretty well because single cell RNA-seq is a sequencing-based application. And would you be interested in interviewing for it? And they were kind of desperate. So I did. And I got the
feeling.
Yeah. It's like, all right. And so I got the job and I jumped, head first into the single cell world, learned as much as I could.
So I guess, again, just for the listeners on a layman's term level, what is single cell?
Yeah. So single cell is acquiring sets of data from individual cells. That's basically it. So think of a cell as a person, right? You can think of it as representations of different data sets, right? So you have like your height, your eye color, your weight, your shoe size, right? And you can take these measurements and build a picture of what a person is, where they're from, how old they might be, right? And you can use those insights to learn something. It's the same thing with cells. With single cell, you can measure different aspects. Now the type of single cell that I'm interested in is measuring gene expression. So you can take one cell and ask the question, what is that cell doing? And to answer that question, as a surrogate, you look at, well, what genes is it making? Because it stands to reason, right? That the cell is making stuff, making genes, making proteins that are necessary for it to survive in its environment, right? So it'd be kind of like asking the question about a human, like, what is their job? Okay, let's look in their backpack. Yeah. What's in their backpack? Oh, look, they have a notebook. They have these pens. The notebook is about X, Y, and Z. Oh, this person must be an engineer, must be a scientist. Oh, but we found this cell in the context of, or a human, in a construction site. Oh, it must be a construction worker or an engineer or something, right? Same thing with cells, right? So, when it comes down to it, you can do all this fancy technical work and explanations and stuff, but that's really what we're doing. We're counting different items from individual cells. In this case, we're doing genes and we're doing proteins. And from those counts, we're able to figure out, what cell it is, what it's doing, what's its functional state. We can use that information to then discover, what are the best treatments, for a particular disease, for cancer or something like that, and really understand at a cellular population level the impact, say, of a given drug is having.
I was going to ask, so this seems like an incredibly powerful technique. It's one that I would say from my own experience is not readily accessible at the moment because of the scale of what it is. But its applications are so varied, right?
Yes.
Is there any application for evolutionary biology? I'm just thinking is there any researchers out there who have kind of used the single cell technique and answered an evolutionary biology question that you know of potentially?
Not off the top of my head, but single cell is everywhere. And that's what's really cool about it. It's like you can go into someone who studies muscles, who studies bones, who studies nerves, and they've either used it or thinking of using it, or someone has published an impactful paper in their field. So I wouldn't be surprised if there wasn't a paper in evolutionary biology that's done so. I mean, with single cell, you can do lineage tracing.
Cool.
So I'm sure someone has thought about it or maybe done an experiment. That's not really my background, so I'm not too certain. Yeah.
I mean, I think, again, going back to the scale of this this technique, the amount of data that you can get, do you foresee that in the future obviously at the moment you look at a publication, or maybe 10 years ago you look at a publication, somebody's looking at one gene, one protein, the structure of that protein in one particular state, one particular disease state, and they've come to a conclusion that way, and they've been able to publish in that respect. But with the advent of single cell, you also, I know, are quite interested in AI and machine learning and that kind of thing. How widespread do you feel single cell analysis is going to become in the next sort of maybe 10 years?
Yeah, so that's a great question. And what we're seeing now, the trajectory has been, going from a few cells, and now people are doing millions of cells, tens of millions of cells. And there's huge global efforts going on to like Human Cell Atlas, for example. Their goal is to map every single cell in the human body. Then you have the Brain Atlas projects, where people are mapping, whole cells in mouse brains, human brains. And so I think within the next 10 years, we're going to see an incredible explosion of tools associated with single cell that help drive the number of cells we're analyzing. But the next evolution, and we're already seeing this, is this application called spatial omics, right? Where it's not just looking at, cells in suspension, right? It's looking at cells in their native as possible in the tissue.
Okay.
So not only do you get, information about who that cell might be, what are they doing, but where are they? And then in relation to that cell, who else is there? And, that's another layer of information that's incredibly helpful. So to give you an example of why that is, there's this concept of in tumors, right? Sometimes you get immune cells that are responsible for killing cells actually become less effective in doing so. And this has to do with something called like immune checkpoints. Basically, the cancer can evolve and figure out a way to turn them off, right? By giving false signals. Well, that's thought to happen because of contact, right? So like those two cells have to physically interact. And we've done a lot of work, I mean, not me, but the field in general, looking at the relationship between certain molecules and how one shuts off one cell or activates another cell. But with spatial, you can actually see those two cells interacting, and you can pinpoint like, hey, look, here's that signal from those two proteins interacting.
Wow.
And that could help us understand, why do tumors not go away on their own, or why does cancer metastasize? And it has to do with how healthy cells are interacting with cancerous cells.
So on the sort of practical scale, what does a sample type look like between single cell analysis and spatial omics? Yeah,
so in spatial omics, you have whole tissue. So typically, researchers will take a piece of either human tissue or mouse tissue or animal tissue. They can do a couple of things. They can flash freeze it and then embed it in some matrix and then slice thin layers and lay it on a microscope slide and then do some work with that and then image that. For single cell, you could take that chunk of tissue and use enzyme to digest it and break up the individual cells, and you lose that spatial context, right? So the samples can be pretty similar. I mean, you could also, in theory, take a piece of the tumor or tissue to do spatial transcriptomics, and then another piece to do single cell or some other whatever you're working on, and So like that's the beauty of these two techniques, right? They kind of feed into each other, and one gives you a lot of information about genes and expression levels and stuff, but it doesn't tell you where these things come from. Whereas the spatial right now, it's a little bit less on the genes, a little bit less on the proteins, but you get that spatial element out of it.
This is a question that I'm not sure is even, it might be a very stupid question, but would you be able to ever do this on live cells or live tissues? Maybe
as it stands right now, I mean, to me, that's like Star Trek, right? Right. That's something I dream of, right? How would you do that? Can you imagine? I mean, I just imagine like having a little camera and just watching biology occur, watching it, right? In vivo, in completely unlabeled, just natural context. I'm an optimist, and so I think it's just a matter of time. But with the current state of the technology, I don't think that's feasible.
Okay.
Yeah.
So again, let's drill down into to the platform and actually the protocol of what a scientist would do in their lab. If let's go with the single cell, they've got their sample. I guess it uses an antibody and a DNA barcode. Maybe you could talk to me about the protocol and how a scientist at the lab bench would actually go through this process.
Certainly. Right. So the area I focus on is single cell RNAseq and multiomics, particularly looking at generating data from single cells that has both genes and proteins and being able to map that information back to individual cells. Mm-hmm. So in that case, what a researcher would do is, whether they're working with whole tissue or blood, right? So white blood cells derived from blood is a common sample type. You would process either your tissue or your sample so that you have a very clean preparation of cells of what's called a single cell suspension. So basically all your cells in a little soup, mm-hmm. right? Mm-hmm. And then for our applications where you're doing proteogenomics or proteotranscriptomics, you would have to label the cells with these antibodies. Now, in this case, those antibodies are special because you have a little DNA barcode attached to them. So historically, antibodies have been used for things like flow cytometry, immunohistochemistry, right? So the very imaging-heavy applications. By putting a DNA barcode on it, though, you get rid of that. I mean, you can do some imaging and stuff, but in this context, that's not what I'm talking about. So you put a DNA barcode, and now you've converted the event of an antibody recognizing its cognate antigen or its little molecule into a sequencing-based readout, which is incredibly powerful because imaging modalities rely on light. So you would have to have some sort of fluorophore attached to your antibody, and if you know anything about flow cytometry or imaging, you have issues like spectral overlap. Hardware considerations that might limit the number of things you can look at the same time. Well, you don't have that anymore with single cell multiomics, or what we're working on called scRNAseq. And scRNAseq stands for cellular indexing of transcriptomes and epitopes by sequencing. Cool. It's just a fancy way of saying you can measure genes and proteins at the same time. Yeah. They both use sequencing. Mm-hmm. So you would take your cells, you would stain them with these antibodies like you would for a flow cytometry experiment. You do some washes. And then you run those labeled cells through a single cell RNA-seq workflow. So the most common one by far is probably a company called Proteintech Genomics. We work with them, we're partners with them. And you can do millions of cells per experiment. And at the end, the researcher would take their labeled cells, run them through this kit. At the very end, they get what's called a count matrix. Remember earlier I said we're counting things. Yeah. So you just get a table that has counts of things. Mm-hmm. And from that, and these are pretty big tables, but they're text files, so they're not huge files. And you can do statistics on them and figure out like, okay, this cell has 8 counts of this protein and 10 counts of this gene. Well, so does this other cell. So they must be closely related, right? So you start grouping them and clustering them together and visualizing that data. So users, will spend maybe a few days to a week doing the wet lab, the staining, preparing, generating sequencing libraries, and then spend weeks, maybe months doing the data analysis. That's really the fun part. Yeah. Where you're doing all the statistical techniques, trying to find patterns we call these data sets highly dimensional because you're counting a lot of things along a lot of dimensions. Yeah. Yeah. And
do you think this would be, I guess, do you use a lot of AI in that? What is the process that you do process that data, and how accessible is that?
So right now, most wet lab or bench researchers are collaborating with a bioinformatician. So someone who's specialized in statistics, writing code, and pulling out those patterns, those insights with you. Obviously, they're being guided by the researcher because they know the biological importance of what they're doing. Bioinformatician typically doesn't, but they know the statistics. They know how stuff works. Mm-hmm. So it's a ongoing process, mm-hmm. Big collaboration across big groups. Now the power of AI, I'm a big proponent of AI. I like to use it wherever I can, really just making my life easier.
Write me this email.
Write me this email, contact these people or tell me about this thing. But eventually, we will end up generating atlases of cells, right? Of whole tissues, of whole organisms. And you can use that data to train AI with it. And the vision is that by doing so and generating these massive atlases, you can then feed it data from experimental conditions and have it try and find patterns that you would not see, right? And that's the kind of dream there.
Definitely. Because
right now, I'm not a bioinformatician, right? But I do a fair amount of that type of work. And it's kind of, trying to find these patterns manually can be, it's very time consuming.
Yeah.
Right. You write a little code, you create a little figure, and then you look at, okay, I looked at this group of cells versus this group of cells. What's the difference? Oh, I found these 5 little genes or 5 proteins that are different between them. What does that mean? So I then start reading papers on these proteins and try and figure out what does that mean to me? Whereas AI, will have instant access to all that information and be able to, assuming it works well and is reliable, it'll spit out answers for you and say, hey, think about, you found this protein here. Well, I did a little literature search and I found that this protein is associated with this type of cancer, and I see that you have these samples in my database and stuff. Maybe, I pulled those samples and did this analysis for you. And here you go. I also wrote your paper for you.
That's the dream, really, isn't it? I think, so in my mind, I mean, I'm away from the lab bench now, but I speak to so many researchers who are using machine learning and AI in tools such as, omics specifically. And in my mind, there's a world in 10 years, hopefully 1 year, where there's a machine learning model that has been able to generate a digital human where you can get a sample type from every single tissue, every single disease state. You run it through potentially a single cell pipeline, a spatial omics pipeline, feed it down lots of machine learning models, and you have this picture of a digital human with so much data in it that obviously there'll be biases, but but sort of like an average of every single person's data set could be within that thing. And it's interesting that I think that would change the more we learn about machine learning and the more we realize its usage and its benefit. I think it will change how sort of core fundamental wet lab researchers design their own experiments as well, because they have to assume that their data is probably going to be used to train a model at some point.
Yeah. And that's what people are already doing with these cell atlases, right? They're looking at it. You can do a lot of in silico work and generate a hypothesis and then take that hypothesis, which is pretty narrowed down, right? And then do some wet lab, right? So certainly, that's an interesting vision and dream, and I hope it comes true. One of the things I think about, and I totally saw this in a Reddit post, so don't judge me too hard, but they were saying
Reddit's getting big. Lab rats is out there.
yeah. But this person had posted and said, there's this movement to try and kind of look at biology as from the, quote unquote tech bro perspective, where it's a matter of finding the right code and generating the right data sets. But this person was saying something along the lines like, but biology's, in a sense, because evolutionarily it's the way it's developed, it's kind of kludgy. It has inefficiencies. It has things that if it was a piece of code, you probably wouldn't want to have or wouldn't have.
Yeah.
Right. So you couldn't, there's a lot of hidden variables or things that we can't even conceive of. And that would make it potentially difficult to accomplish something like that.
We're just noisy data.
Yeah, we're just noisy data. But maybe we just don't have the right AI yet to do that, or haven't trained, and I mean, we're just getting started with this, right? So who knows where this is going to lead to.
What do you think is the next sort of 5 years for your specific space, so spatial omics and single cell work? What's the developments that you see are happening? Yeah,
so I think is a couple things, right? One is, although single cell has been now around for a while, right? So maybe a decade or so, or a little less than a decade. We've just scratched the surface in terms of the total number of cells that are out there, right? We haven't completed our atlases. So I think over the next, 4 or 5 years, we're gonna have enough data to really start asking interesting questions, perhaps about evolutionary biology, perhaps about, systemic disease, or more complicated diseases that are multifocal diseases like diabetes, cancer, and stuff, where you just don't have one thing happening and causing a disease, and you can model those things and you can ask questions and design experiments to address more complex questions. But I think the next phase will be the movement of these technologies into more clinical applications, right? And actually, okay, now we've done this work. Now let's start applying it to human health. Right? Can we take a chunk of tissue and identify a single cell of cancer in it and then do something about it? Or can we, and we see this now in genomics where people are taking blood and finding evidence of cancer directly from cancer DNA or DNA from cancerous cells that's floating around in your blood.
Circulating.
Right. Yeah. Circulating. Yeah. Right. So then the question is, like, you have these noisy data sets. We'll have the tools potentially to denoise them and give us insights that'll help us treat disease or come out with better treatments. I think, to me, like, that's a big goal of the life sciences, right?
Could you have it as like I'm going to take this biopsy from this patient at 10 o'clock in the morning. I'm going to send it to the lab in the hospital. They run the system, and by,
trust me, there are a lot of people trying to do just that.
Really?
Right. Just developing the technology, the techniques. Right now, especially in single cell, like the wet lab portion of it has improved a lot. We've gone from a few thousand cells to millions of cells, but there's still a lot of challenges, right? And those challenges are complex. They dealing with sample preparation. What samples are actually amenable to processing with these different techniques? The costs associated with it, right? They're newer technology, so things are expensive, right? And there's a lot of kludginess in the analysis side of things, right? Because it's very open source. Scientists love open source. So you have tools developed here, tools developed there, and they all kind of do the same thing, but in different ways, or one works better, or one is cited more in papers. So people go to, it's just like, we still need to coalesce around that and have like tools and processes that are going to enable, your everyday scientist who isn't a bioinformatician or a biostatistician and stuff, to be able to design an experiment and go from sample prep to data analysis in a very short amount of time.
Yeah.
So that's kind of one of the big challenges with single cell. It's a big village effort right now. And I think the next phase is simplifying it enough. So, like, for example, sequencing. When I started, we had an instrument called the GA2X, the Genome Analyzer. And I remember, people calling me to troubleshoot it, and I'd remote into their computer, and it was very manual. You're trying to align like optical systems and clicking around and trying to do this, right? And it was a bit of an art. You had to know enough science, but then sometimes like knowing where to look to find the problem.
Yeah.
Right. And now, like, that's all gone, right? All of that's automated in a system. Most of these genome sequencers, it's like you do minimal sample prep, you load it onto a machine and hit a few buttons. Your data gets generated, goes into the cloud. And, and the company I work for, Illumina, had this, push button bioinformatics, right? Like, you just gave it your data, push a few buttons, and you got answers. Mm-hmm. And like, that's kind of where we want to go, right? That's going to open up this technology to the most number of people. And we're just not there yet.
I'm going to ask an ethics question.
Sure.
Do you foresee, obviously, with the amount of data that we are going to generate from human samples, predominantly, obviously, in the healthcare sector, if we are going to move into the healthcare sector with this sort of technology, do you foresee any sort of data restrictions or ethical restrictions that are going to stop the development of the technique? What do you think? What are your thoughts on that?
I don't think these types of concerns will stop the technique, right? Because especially around like human samples for medical reasons or processing for medical reasons, there are a lot of safeguards, regulations around, right? In the States, we have like HIPAA, which prevents, basically anyone from discussing any private information. It's all on a need to know basis.
Right.
I used to work in a pharmacy, right? And back when I worked, I'd be at the hospital and I'd see an interesting order come in, and it was like a certain drug. And I knew that drug was used for spinal injuries. So I'd look up that patient and say, oh, wow, 19 years old, motorcycle accident, and, there were no regulations back then, right? So it was just me learning about this. And, and then HIPAA came, and it's like, no, you can't do that anymore. You have to just, in that case, put in your order, send the meds, and ask no questions because you don't need to know.
Yeah.
Right. So there's a lot of regulation around that, especially in Europe, right? UK, there's sensitivities around the ethics. And I think in part because of sequencing.
Yeah.
Because, you could, in theory, if an insurance company were to sequence you before you were sick and they knew, well, you have a propensity for heart disease, we're not going to cover you. It's a pre-existing condition or something, especially in the States, right? I don't know if that happens.
I guess, are these things going to become like protected characteristics, I guess, if your genome becomes a protected characteristic that you can't, discriminate against in HR terms or that kind of thing?
Yeah. I think that's something that will happen, is happening, right? Because you can't discriminate based on your genetic profile.
Interesting.
There's a great film from the late 90s called Gattaca.
Oh, great film.
I love that movie. And it tackles these ideas, right? In a very cool and very engaging way. One of my favorite movies, hands down.
I actually remember watching that at school to ask these questions. So I think that we're talking about like invalids and valids, depending on like whether
valid
they've been
invalid,
genetically engineered humans and that kind of stuff. Which is obviously not what you're aiming to do with your research at the moment. So I guess another question would be let's circle back to your PhD times and pose the question, if you had the technology that you have now, but you were doing the research of your PhD, what would be the experiment that you would run? Oh, yeah. So, just to give you, this is the experiments I would do, right? So I would work with mice and they were a model for Salmonella infections. So you would infect the mice, and these would be transgenic mice. Maybe they had some genes knocked out. And one of the big questions I had at the time was, what are the cells that are present in the mouse intestine, right, as a surrogate for the human intestine? And what are they doing, right? So I had to design flow cytometry experiments and do these very tedious and experiments that took a day and a half, right? No sleep, just brutal working through it, preparing multiple samples because you'd have to weigh all the tissues, homogenize them, plate them for bacteria. You had to extract protein, you had to extract RNA, DNA. I had to genotype my mice to make sure they were actually knockouts, because sometimes you'd buy a mouse and they weren't knockouts and you'd get weird results. So the experiment I would do now is a very simple single cell RNA-seq experiment where I would just take all my cells, don't even need to label them, although I would use DNA barcoded antibodies now. But if I could even just do the RNA part of it, it would show me in one experiment, or probably 2 or 3 experiments, show me what are the cells that are present and literally what are they doing, right? But that would kind of, I kind of made fun of my PI because he would just have to sequence one gene or and it took him 4 years to do it. It wasn't easy, right? So now my PhD would now be like a 6-month project, Yeah, and I'd have to follow that up. But yeah, I would love to take human intestinal tissue, mouse tissue, whatever model we're working with, whether it's disease, HIV or cancerous, and just understand who's there, what are they doing, and why are they doing it? And I think from those insights, you can derive, potential strategies to develop therapeutics, and really just help people. I mean, that's I think at the core for me, that's always been an important aspect of it. Whether it's solving their problems or, providing a little insight into some biological problem, that's a big part. Definitely. Big
low-lying, part of my drive.
Yeah. It's interesting to kind of think about, like you said, the experiments that you could have done now would shorten your PhD to a 6-month kind of timeline. And it was the same for me during my PhD. I got to the end and then AlphaFold was developed and then basically I put my sequence of my protein into AlphaFold and it popped out the structure that I'd just worked 3 years to solve. Yep. And that was a moment where I was like, okay. But it's kind of a nice highlight of the development of biology and research, and not just biology, chemistry, physics, biophysics, everything in between. And it's very interesting to look to the future and think, okay, we're on this massively exponential increase in the technologies that are coming out and how they're being used and the different disciplines that they're being used in. And it's kind of scary to think about, in 10 years time, what are we going to be looking at? Do you have any thoughts of a new thing that we wouldn't even think about right now?
Yeah. So one of the challenges we had, and I think this is something that still happens, is the variability between human beings, right? You can get, and I see this in our work now. We use donor blood. So we'll purchase it from a company, and these companies go and they pay people for their blood, and they isolate white blood cells. We do experiments with these, and we see this incredible variation in responses from time to time, right? Some donors will have, 100-fold higher of a specific type of cell than another one, right? So like you can develop a mouse model, and they're all inbred, right? So they all have pretty similar genetic backgrounds and stuff, and that's helpful. Obviously, mice in that type of work have an indelible legacy, and you can't change that. But accounting for the human variability, I think, is going to be, another big phase of it. And because ultimately, if we don't solve that challenge or that problem, a lot of the insights we derive from the current work we do are going to be here or there, right?
Yeah.
Not applicable. They're just snapshots of random. So now I think with these higher throughput or tools and AI, right, we can now start integrating, across, I hate to say it, but wild type humans, and and try and assess what is actually going on. What is your typical human? What is that model human, right?
Yeah.
How do you do that? And it's kind of a model of models, and I think I think
there'll be multiple. It'll be so interesting. I'm going back to evolutionary biology as well. We might actually find, different subspecies and that kind of thing, and how we respond differently to environmental cues and everything in between.
I think that challenging a lot of our presumptions or assumptions that we have about what it means to be human.
That's really exciting. Yeah. That's such an exciting field to be in.
It is.
Ivan, thank you so much for this conversation.
It's been a pleasure.
I've really, really loved it. And I'm sure our listeners will have a bit more of an insight into the world of genomics. And I'm going to say, if they want to get in touch with you for any collaborations, please do.
Please do.
Thank you very much.
Thank you for having me.
So to our listeners, if you enjoyed this episode today and would like to learn more about the big questions that help us to understand the book of life, then like and subscribe to Proteintech's podcast. Thank you for listening.